What a great way to start the afternoon session, another story of hope and inspiration. So I'm really excited to hear the talks in this session, and I'm glad I stayed, and I'm so glad everyone has stayed for this afternoon session on artificial intelligence and machine learning. It's very difficult to go anywhere without hearing these terms, and clearly these approaches and technologies are making a huge difference in our lives already. All kinds of useful things, from college students getting help to write up their midterms in time, extending all the way to things that will impact patients here very soon and are already impacting them, I would say. A drug called RLY-4008 that many investigators here may have worked on was actually discovered largely through AI and machine learning-based approaches. So to moderate this session, I'd like to introduce Dr. James Cleary. He's a medical oncologist at Dana-Farber Cancer Institute. I learned over lunch that we share the same alma mater. He went to Rutgers College, where I did medical school. This was subsequently followed by medical training at Albert Einstein, MGH, and he's now at Dana-Farber. Dr. Skye Mayo is joining him to moderate this session. He's surgeon extraordinaire at Oregon Health Sciences University, and he's got a fan base here too, I see. Okay, so I'll leave both of these tremendously talented gentlemen to take over this session and look forward to hearing the talks.
Thank you. Thank you for that great introduction. And thank you to all of you for staying for this last session, because I agree, this is a really exciting area. And I think really in medicine, the time is right to start using these technologies. So our first talk is going to be given by Dr. Julius Chapiro on using AI in radiology.
Thank you very much for the introduction. Thank you very much for having me and for welcoming me here. This is a highly energized and extremely optimistic meeting, and I'm really enjoying being here. I'm here not only as an imager and a data scientist, I'm here also as an interventional radiologist, and it is my firm conviction that the sooner you involve an interventional radiology specialist into the care of your cholangiocarcinoma patient, the higher the quality of life and the better the outcomes will be. So I just want to advocate for our presence here at this meeting and in the care of these patients. So my task was to talk about the applications of AI to imaging and intervention. And I looked at it and I wondered, how hot of a topic is this in cholangiocarcinoma? And obviously, you know, cholangiocarcinoma research as a whole is pretty hot. You have, if you look at the publications, there's a lot, and it's increasing in amount, 21,000 papers published. But if you go and put in artificial intelligence or machine learning or deep learning, you really have very, very few papers. And I updated the slide from a similar talk that I gave last year, and only 25 papers were added. And the question is, of course, why? And I'm going to try to tackle this from an imager's perspective. So I'm dividing this in 4 fields: the problem itself, the technology, the vision application, and the academic-industrial partnership, which I think is very important. So let's start with the problem. So the basis is that we have an overwhelming and a very rapidly growing amount of data. We all have seen in this meeting, and particularly in the very encouraging sessions in this morning, how we are about to harness a lot of the data. And Stanford Medicine predicted that the data between 2013 and 2020 is going to multiply by 10-fold or 15-fold. In reality, with the introduction of data science, it was 1,000-fold. And you see this typical table of patient characteristics that is published with every paper. It's a lot of data. I call it data trash because very often it just characterizes but really doesn't give you any useful information about how to use it clinically. So every cholangiocarcinoma patient undergoes a multimodality imaging journey, right? For example, in my patients, the local regionals, some of them have ultrasound screening, some of them become apparent clinically. They have a baseline MRI or MRCP where we diagnose this, and then we have interprocedural imaging, whether you're doing local regional therapy through the artery like TACE or Y90 or others. Then you have multi-detector CT, and then you have follow-up MRIs, numerous scans. All of them obtain a large amount of data with multiple sequences, and all of this is poorly integrated. So this is a significant loss of information. Those are the liver cancer diagnostic imaging guidelines, and when there's 25 of something, and that includes cholangiocarcinoma, that means nothing is really good. So all of these have unclear, varying, and inconsistent definitions of imaging features. For cholangiocarcinoma, we obviously need biopsy in nearly all tumors. Most references and most guidelines are based on retrospective data, and we have varying, contradicting, or simply no recommendations how to properly assess tumor response. In light of all these new drugs that are coming on, that's a real issue. And obviously, we have different decision trees for therapy allocation depending on the tumor location and the tumor subtype, and we have very heterogeneous concepts on how to approach therapeutic strategies. Then for my field, we can cook, boil, freeze, and electrocute a tumor in various ways. We can chemoembolize it, we can put drug-eluting beads in, or we can try to kill it with radiation. All these varying different techniques cause very different imaging appearances. And then we have very complex genomics and proteomics. You heard a lot about this. Variety of different tumors have very vastly different profiles. The only common thing really in cholangiocarcinoma is that we know that they originate from the same type of cells, but these tumors are vastly different in terms of their genomics and proteomics. Because of that, we have numerous molecular targets. So you're getting the picture. It's really chaotic. It's a lot of data, and we really have not a really good understanding of how to deal with it. And all of these molecular targets produce different drug mechanisms and action that cause different imaging appearances, right? And so because of that, all of these novel agents that we're introducing are formidable imaging challenges. Also, another major challenge, and this is a summary for most of you, this is a rare disease with limited central data that is managed, and I'm very, very gratified to hear that the Cholangiocarcinoma Foundation is doing so much to centralize this data. So the problem was that we have an overwhelming amount of complex but poorly organized data that may impede effective clinical practice. Now, a few words on the technology. I'm not going to go too much into detail, but I think it's my obligation to just explain this a little bit in layman terms. So we have this burden of data in electronic health records, genomic sequencing information, reporting that can be tackled with natural language processing. And then we have computational power at a very low cost right now. And all that meets this whole realm of AI or different subtypes of machine learning or data-driven learning, such as deep learning. And so the original promise was that all this will do is to deliver workflow efficiency, diagnostic accuracy, and precision medicine to us. And so we've looked at a couple of aspects where we can use this, from diagnosis to follow-up imaging, to automate diagnosis, to improve outcome prediction, to make sure that we have the proper therapy decision, and then to measure tumor response. And I'm not going to go into detail to that because there is a separate lecture on that, but we also understand now that this technology can be used for drug identification and can also be even used for antibiotics discovery now. So, but taking a step back, this technology is not new. It has been used by the US Postal Services in the 1970s for the first time, and if you look at these digits, they used it to classify and characterize digits on an envelope. This is 504192 for you, but when you see the lower digits, then you have to twist your head a little bit to recognize it as a 4 and a 2. But in essence, this is a pattern recognition. And so for a machine, that's not as hard as for us. Therefore, we need to train it with a lot of organized input data that has pretty homogenized and a high level of annotation. And then we identify patterns in this data and then train the algorithm. And then what we do is we feed it with an additional new set of images and then can see and recognize in the imaging data sets things that we haven't seen with a bare eye. And there are different types of workflows in this data, and we can apply these mechanisms to, for example, liver segmentation, tumor classification, prognostication, depending on what we apply it for. Now, it's very important when we look at data to understand how to pair the type of data with the appropriate machine learning algorithm, because we have obviously just generally numeric unstructured text data, we have tabular data, but we also have images. And so these specific types of data must be brought together with a very specific types of algorithm, and that's where we are starting to learn and to understand how to best handle these things and how to combine these types of data. Now, when you start training a neural network, basically at the beginning you have this nascent neural network with multiple layers. You have an input and you have an output. And in the beginning you start training it with a piece of data, and the output is pretty bad. It doesn't really segment this tumor. Then you start tweaking the layers one by one, more and more, and then you have a reasonable result. After you do this 10,000 times, you have a fully trained algorithm. And then you have this trained network, and you can feed it with new data as never seen before, and then it can make a really good prediction to, for example, to infer for whether this is a malignant HCC, malignant non-HCC like cholangiocarcinoma, or a benign tumor in the liver. And the vision is to make sure that we have a fully automated, fast and reproducible, reliable characterization of liver cancers, including cholangiocarcinoma, based on this work. Now let's talk a little bit about application. So our group looked at automated detection and delineation of liver lesions, for example, hepatocellular carcinoma on the MRIs. And it's important because we know very often these things are overlooked in a patient who had cholangiocarcinoma. Very often you see the first diagnostic scan where you actually know this is there, but then you go back and you kind of can see it, but it was not exactly diagnosed. And so it's important that we look at early detection and really identify subgroups on imaging where we can just screen patients. And then obviously once you have this tumor, you want to have automated diagnosis, and we've demonstrated that machine learning driven image analysis can provide us with very, very high quality automated classification of lesions in milliseconds, and as we have a large amount of patients to go through, that will be helpful. And in this particular case, we trained a machine to really do that in over 500 lesions and tested it with 60 hepatic lesions and demonstrated that the neural network actually beat 2 radiologists with extreme experience, even specifically in the cohort with cholangiocarcinoma. So we are already experimentally there. We now are working on red path validation. As you know, some of the HCC can be diagnosed with just imaging, but some of it can't, and I think that's the case for cholangiocarcinoma for most of these tumors. But the question is now, can we apply imaging, deep learning analysis in order to diagnose this disease without invasion? And that's possible, I think, in the realm of possible, and we just need to feed the algorithms with data. Another important concern is the explainability. Right now, we're considering all these algorithms to be a black hole. So you put something in, it puts something out, but we don't really know what's in there. So how do we introduce explainability? And we had some experimentation done in our group, and other groups have reproduced it, where we sort of disentangle the decision-making of the algorithm and can have a prototype that highlights the features using a heat map on the images to show to us, why a particular tumor is a cholangiocarcinoma or is not a cholangiocarcinoma. So those are the topics that we've been handling. And then ultimately, we have recently worked on the ability to predict response to a certain therapy. This was a very small cohort where we used age, gender, tumor type, comorbidities, baseline imaging appearance, and fed this information into a neural net and then predicted whether or not this patient is going to be a responder to local regional therapy or not. And turns out that we can do it with a nearly 80% accuracy in a very small cohort back then. This paper was cited over 150 times, so that shows you really how much interest there is in the wider liver community, and certainly this can apply to cholangiocarcinoma. We also looked at the ability of these networks to help us predict disease recurrence, for example, after early stage liver cancer, resection or transplantation or ablative therapies, and have demonstrated that the best way to do it is to combine both imaging data and clinical information of the patient, such as laboratory parameters, to predict whether or not the tumor will recur. Now, an interesting field where we're looking at right now, and this is beginning to be widely studied, is differentiating HCC from mixed tumors with ICC from ICC tumors. And for some reason, that is a classification problem that is probably the first priority right now for us to tackle. And so a lot of people are trying to do that. They're using ultrasound images. They're using contrast-enhanced ultrasound. For example, this group really was able to demonstrate fairly high predictability, being able to distinguish different types of these tumors and to subject them to the correct treatment. Not going to go too much into the pathology, but I felt that this paper was very, very important. I was actually the handling editor for this, and this was published. So this group really demonstrated that we can use deep learning for classification of different subtypes of cholangiocarcinoma on tumor biopsies, and digital pathology. You're going to hear all about it. This is really a hot field, and again, it's being reproduced. This group from France and Germany really demonstrated that differentiation of tumor biopsies can be handled, and in the age of automated digital pathology, we can bring rad path studies where we can compare imaging and pathology and try to deduct how the pathology is going to look like based on the imaging appearance. That's a hugely interesting field to us. And obviously, also we can use machine learning to identify, for example, novel biomarkers using proteomics and screening in peripheral blood and bile itself on this topic. And our group, for example, right now is screening aspirates from biliary drainage catheters for biomarkers, and then we're feeding this data into our machine learning algorithm. And then again, similar true is for genomic mutations. As I demonstrated, these tumors are complex, very heterogeneous, and so I think it's important to look at these applications in this realm. Just briefly to mention, evidence-based medicine is usually hypothesis-driven, has low data point diversity, and we're comparing between groups. In machine learning, we are data-driven, we're having a high diversity of variables, and we're comparing between variables, and that makes it so strong and powerful. At the end of the day, what's true for EBM and machine learning is that confidence will only be reached if it's reproducible, and that's true for both. So putting it all together, I think the past was intuitive medicine, the present is evidence-based medicine, and the future is algorithmic-driven, precision-guided medicine. Where do we stand? I think we're just starting. This is my favorite rule. I believe we have a lot of interesting initial data, put in some 20% of the effort, got 80% of the data, but we need to put in 80% more effort to get the remaining valuable 20% of all the data to make sense of it, right? Implementation in clinical workflow will certainly work in the background where we add, for example, to the tumor board these clinical informations and decision-making support systems with quantitative imaging, and they will help us make the better decisions for our patients. And then ultimately, I think the liver community must do more to engage in high-quality AI research. I call it not from bench to bedside, but from code to bedside, and I think we need to do some work introducing this into the cholangiocarcinoma Foundation. Just a few more words on the academic-industrial partnership, and then I'm going to finish. So most important thing when you build these things into a new field is that you need to build a compelling value proposition. So AI can't solve every problem. Not every investigator has a good product. So don't create solutions in the absence of problems and focus on the blatant critical issues rather than aspirational and latent. And do it not in a black box between engineers and physicians. Have iterative processes involved, and that's where I think the success lies. There was consolidation in the players in the field in radiology. I'm not going to bore you with that. We're past the plateau of just development. We're now reaching a very productive level where we're engaging with groups and communities such as the Cholangiocarcinoma Foundation, and now are reaching a productive level where we actually are beyond the hype and overpromises. This is disruptive technology. It will be regulated by the FDA. We're thinking about this in many ways, and before deployment, there's going to be some ways how to really propose a regulatory framework for modification of this technology, but we're not there yet. There aren't many products that outside of basic imaging applications are actually applying to cholangiocarcinoma. Be aware of benchmarks for quality for AI research. Not everything that says AI is actually cool. Some of it is just blatantly just using the term to just land a paper. So we need to look at that very carefully. It's unclear who's going to pay for it and how it's going to be done, but I encourage industry to really invest in this effort in the Cholangiocarcinoma Foundation and to participate in an effort of data collection to afford us this kind of research. So conclusion slide, it's about the data. The key issues are that we use and transfer large amounts of raw data between academic institutions and societies, and cholangiocarcinoma is not very well organized. And I think that the Cholangiocarcinoma Foundation has a phenomenal responsibility and opportunity to collect this data. We need large amounts of high-quality, annotated, curated data. And a very specific, narrow goal of what we're going to do with that. And I think that this is the community where we can definitely achieve that goal if we tightly calibrate and invest. All right. The next big things are going to be personalized staging systems, imaging of response, and digital pathology. You're going to hear all about it in just a few minutes. And with that, I close and thank everybody for the attention. Sorry for being a little over time.
Thank you so much, Dr. Chapiro, for providing a, I think, really insightful and inspirational foundation that's going to work well for the rest of the talks to follow. Our next speaker is Dr. Benjamin Goeppert. He's a professor of pathology at RKH Hospital in Ludwigsburg, Germany. Dr. Goeppert.
So thank you for this kind introduction, and thank you, the organizers, for having me and putting up this amazing conference. I would like to share with you now, slowing down a little bit in tempo, the artificial intelligence in diagnostic pathology and especially with cholangiocarcinoma. I'm a clinical pathologist. I worked roughly 15 years now on cholangiocarcinoma, and I got into AI just recently through a young colleague of mine, and you will see some projects of us just in a second. If it's working. Yeah. So the next 50 minutes will be a short introduction. I thought maybe a little bit longer introduction to biliary tract cancer and the morphomolecular state of the art. And I will show you in a second why I think this is important to know. And then how AI would be of any help in doing this. And then, of course, open tasks and how we can approach them. To date, where do we come from? Where do we stand? What is needed? I show you here that resection and resectability, as we heard before, is really crucial in this disease. You see here's patients, a patient collection that I collected in my time in Heidelberg, and you see that complete resection is fundamental and plays a huge role in prognosis of the patients. And surgery is still the only curative therapeutic approach for BTC patients. However, only a lot of patients, as we heard just a few minutes ago, are not amenable to surgery because of their stage being detected with this disease. So predictive diagnostic pathology is of high importance for these patients. So a quick and reliable diagnosis is required, and a colleague of mine from the US, Vikram Deshpande, told me it's a huge problem in the US as well, that the diagnosis is delayed because the pathologist and the clinicians together, they do not come up with a differential diagnosis of cholangiocarcinoma. And another important feature is that if you get a biopsy, the material should be preserved for additional molecular diagnostics, as you heard before. This is taken from a rather ancient historical review from colleague Greg Gores, but for me it's still very useful because it strengthens that you have to evaluate from a metastatic disease. So you have to rule out together with the clinicians whether it is a metastasis or whether it is a primary liver cancer, in particular cholangiocarcinoma in our field now. And clinical markers are not histological markers. I will come to that in a second. And when you know it's a cholangiocarcinoma, then you have to define whether it's an extrahepatic type, of course, or intrahepatic type. And if you know that it's intrahepatic, then you should know whether it's a small or a large duct intrahepatic cholangiocarcinoma. All roads lead to Rome, but the diagnosis for cholangiocarcinoma, as this slide is quite busy, can be really a lot of work and sometimes not coming to an end very quickly. And it used to be for the last decades a diagnosis of exclusion. So the pathologist would do a lot of tests to prove or to rule out that it's a metastasis. And at the end, he would say, no, okay, this adenocarcinoma of pancreatobiliary type, rule out diagnosis, maybe it's a cholangiocarcinoma. And if the clinician is not working well together with the pathologist, this would delay diagnosis. And of course, the tissue biopsy would be lost because of the use of many immunohistochemical stains. We are changing this at the moment a little bit because it's coming towards a positive diagnosis. So we have new tools like CRP or albumin in situ hybridization and N-caterine. And together we just recently showed that this is a useful tool to come much faster to a positive diagnosis of cholangiocarcinoma. Another possibility is the use of DNA methylation, and we heard this before. And this could be really nicely shown by a colleague in Berlin. I used to work together with David Kappa, and they showed in several large cohorts that it is of high use in discriminating intrahepatic cholangiocarcinoma from ductal adenocarcinomas of the pancreas and other tumor types. And we've shown before that it's also useful to do subtyping in intrahepatic cholangiocarcinoma. So coming back to histology as the basis for AI, you are certainly aware that biliary tract cancer are predominantly adenocarcinomas, and they have a broad histologic and immunohistologic variability. And this was the problem before because there was no positive marker for these tumors. But it's very important to know that roughly 5 different subtypes that are listed here make up over 90% of these tumors. And noteworthy, prognostic value of simple histosubtyping has been known for decades. This is data from 1990, so really old data, and they showed at that time that papillary phenotype had a better prognostic so a better patient outcome, so was significant from the prognostic point of view. We later could learn that this is most often associated with PSC, biliary tract cancer, and with IPNB, which is a precursor lesion of the biliary tract. Coming now closer to the main goal, we are now at the moment in the intrahepatic cholangiocarcinoma in the status that we can define different subtypes. And this paper set the tone for this because it published already in 2014 that we have different subtypes in intrahepatic cholangiocarcinoma, which was called the cholangiolar type and the bile duct type, which corresponds to the WHO small duct type and large duct type. And I said here, quite bold statement, it's a misnomer, but I will explain why I'm of that opinion, because if you see on the right, oh sorry, you're coming back, yeah. You can see here, this is not a small duct, this is a quite large duct. So, and the ducts are not, the size is not defined at all. So this is often why non-experts are thinking, oh, okay, this must be a large duct, but it's at the end it's a small duct intrahepatic cholangiocarcinoma. And the other way around, with a large duct type, you have small ducts here as well. So this is hard to comprehend for non-experts. So there is a need for clarifying. And this culminated in a table, and I have a modified version here from the WHO classification. And you see, it's not only, it's called small duct, but you have several features, as you can see here, the 5 features who are playing a role and are equally valid to make up the subclassification. So tackling the first task to differentiate intrahepatic cholangiocarcinoma from metastases. This is a very frequent scenario in the clinic, and most often it's a metastasis, you have to say, because intrahepatic cholangiocarcinoma is a rare disease, and metastasis in the liver is a quite frequent phenomenon. And we came up with this publication in 2023, where we could show that it's possible to discriminate intrahepatic cholangiocarcinoma from colorectal metastasis. And this would have certain advantages because you have no loss of material at all, and you can use it for molecular diagnosis at the end. The H&E scenario is like you can see here on the, I won't bother you and test you which is which. The left is a metastasis of colorectal, it's a metastasis of colorectal cancer, and you can see, as the pathologist refers to, a dirty necrosis and the typical cribriform, so back-to-back glands, and desmoplastic stroma as well. But the cells are a little bit different. They are more cylindrical, not cuboidal. And on your right is an intrahepatic cholangiocarcinoma. And there you can see the more cuboidal cells and a desmoplastic stroma and infiltrating lymphocytes. Of course, you have to train this, and we did this with resection specimens and biopsies. And after training, we got tremendous results, in my opinion. The prediction accuracy was over 96% without any immunohistochemistry or so, just AI, of course, by an H&E slide, so the conventional slide the pathologist uses. And you can see what my colleague just says. We want to know what the computer does, of course, and how it works. And we could explain here why he's often telling us the correct diagnosis. He looks, funnily, at the same things a trained pathologist would look at. He looks at the epithelium, looks at cribriform architecture, and intraluminal necrosis, so to speak, the dirty necrosis. And so he does quite the same as we do when we received good training. And then we did a reader study, so to translate it into the clinic, how would this work? And we did this with resident pathologists and senior pathologists, and as you can see, it gives a boost in particular to the residents. So the untrained pathologist, or you could translate this to the non-expert HBB, hepatopancreatobiliary pathologist, who by AI will go to the expert field. And this is quite neat, I would say. So the next task would be subtyping of intrahepatic cholangiocarcinoma, and I just want to briefly show you this work which is in submission right now, and we did or we created or put up a huge cohort from 3 large German centers who are concentrating on this disease, and we could show same or equal results, that it's possible to discriminate, and it's looking at features the pathologist would look as well. And this has huge implications because, as you know, the intrahepatic cholangiocarcinoma small duct type is correlated to the druggable targets like FGFR2 or IDH mutations. And therefore, we can say this would be quite useful for the clinic. So what open tasks are there? And you can see I have the same paper as my colleague here from the French group, the Julius Chapiro, who is a really nice colleague from Paris. He's a surgical pathologist as well. And he used it not only to test for the combined HCC cholangiocarcinoma, but he really could reclassify this tumor. So this is a very neat thing, and I think we can learn a lot from AI at the moment. And the other approach, of course, would be that we really test for druggable targets, not only for these I just showed you, but others as well. You could just imagine like HER2, where you could test this or train this sample set as well. And of course, we should, like for software, it's common, you should do more updates. Yeah? So, of course, if you have something new, you should apply this again. And, but the important thing for AI is at the moment you cannot transfer anything to something else. You always have to test new. How can these tasks be tackled? Of course, I would say through collaborative and interdisciplinary research of experts, and there the European Network and the Cholangiocarcinoma Foundation and PSC partners and so on and so forth are of huge value. And I think we should think outside the box, new ways and methods. And I just show you a publication from the Carter group from Germany with swarm learning intelligence for decentralized artificial histopathology. And this is a really new method, and I think we should use these methods to go ahead. So take-home message, we have AI pros, of course a high reliability, the quickness, tissue sparing is very important, I would say. Potential to screen for druggable targets, capacity to outperform even expert histopathologists, and this is true. If it's, of course it has to be validated, but the capacity is there. And we have some cons, of course. The availability of digital pathology, at least in Europe, is not too good, I would say. It has, at the moment, a research use only status, at least for BTC. It's time consuming at the moment. And of course, there are more studies needed. So taken together, helpful tools, as you can see it here, and I explained it to you. And of course, you should refer to an expert center. And using of the different tools, it's always in combination. So still, for the clinic at the moment, I would always recommend that you do an interdisciplinary diagnosis. And at the end, clinicians reading pathology reports. So lost in translation is, as always, very, very crucial. So I want to acknowledge all the people who did the work with me, and I'm really blessed to work with such smart and nice people in combination, and you, of course, and I thank you for your attention, and I'm happy to take questions.
We're going to take.
Thank you. That was a great talk. For our last talk of this session, I'm very excited to hear this. This is going to be using artificial intelligence in drug discovery, and this is going to be by Dr. Mrinal Shekhar from the Borad Institute in Cambridge, Massachusetts.
Hi, I'm Mrinal Shekhar, and I work at Center for Development of Therapeutics at Borad Institute, and I essentially work in very early stage drug discovery, so essentially trying to find molecules or drugs for a particular target. And this conference has been an eye-opener for me because I've never interacted with any clinician or any end user of these drugs. So thank you for inviting me for the talk. So today I'm going to talk about, the agenda would be talking about how do you find molecules for a particular target, how do you identify these, how do you basically improve the binding affinity of these molecules, and finally I'll also talk about certain other techniques where we are trying to do some basic science to understand disease resistance mutations for a particular target. So drug discovery essentially is a slow process which has a lot of failures, single point failures in its path. And as a result, what actually happens is the cost of this drug discovery program is probably increasing over the years. So currently, based on some rough estimates, it takes almost 10 years for a molecule to get into clinic, and it can take almost $2.5 to $3 billion to generate a single drug molecule for a particular target. So can we do something on this? So based on some health economic data, if you can think about it, if you reduce the cost of doing this drug discovery program, making the molecules better, efficient, as well as fast, even by 20%, that can reduce the cost of doing this entire process by almost like $100 million at the start of the drug discovery process itself. And that means that we'll have much more hits at the goal. And that's where some of these computational technique or computer-aided drug design can have an impact. And this is not new. So essentially, we are at the third wave of these computer-aided drug design, starting at 1980s, where you had a cover of Fortune magazine saying that the drug molecules can be made on computers to now, where we are in the age of NVIDIA and graphical processing units, the GPUs, and you'll see these kind of news items always about machine learning or AI trying to revolutionize the drug discovery. So what's different today as compared to what was there 30 years ago? One major difference has been that the cost of doing this computation has been drastically decreased. So you can do much more computation today as you could have done 30 years ago. And secondly, the quality of data is also improving. So as my colleagues had pointed out about what machine learning, so I try to give a brief overview about what machine learning actually is. So machine learning essentially comes under an umbrella of artificial intelligence, and there are different flavors to it, or different kinds of model. In a bigger picture, what you are trying to do is you're trying to have a data, you're trying to learn some patterns in a data to make some predictions on a new set of data, which you show to this model. So how does that relate to a drug or a small molecule? So on the bottom half, you have a structure or how a drug should look like, and we convert that structure to a format which can be a vector, it can be a graph, or it can be a matrix, which a computer can understand. And that is fed to these machine learning models, and these models try to understand the patterns within these chemical structure and try to correlate it to a certain property. So once you have trained these models, what you can do is you give a new structure of a molecule to it, and you to try to predict a property. And the property can be, is the molecule stable or not? Is the drug molecule stable or not? Can it go through the cell or not? Can it be efficacious or not? Can it be soluble or not? And so on and so forth. The other part of computer-aided drug design is the first principle, where we do not work with data, but we're trying to understand certain properties of interactions of a drug with its receptor or its target. And here we use Newton's equations of motion and run these calculations on large supercomputers to pinpoint what are the key interactions which a drug molecule is making to your target of interest, and can we modulate those interactions to have a better drug-like molecule. These calculations are complicated, take time, but can provide you inference. It can essentially tell you what exactly is happening. So a computer-aided drug design team will use both machine learning as well as these physics-based methods in conjunction while we are designing molecules in our program. So the entire, the way we work in the early stage drug discovery, it's a very collaborative process. And if we can divide the entire process into 3 phases. The hit finding phase, where we are trying to find a molecule which can attach itself to a target of interest to modulate its property. The second stage is we take that hit and develop it into a format where the modulation is more efficacious. And the third is to improve its pharmacokinetic property and make it more drug-like. So these 3 stages go one after another, and it's very collaborative. So what do I mean by the hit finding stage? So in the hit finding stage, let's say we have a receptor. And this technique is called virtual screen. So what we do is we computationally screen a library of, say, 100 million molecules against this receptor. And by screening, I mean is we take each individual molecule in our database, and we put it inside a receptor, we rotate it and translate it, and try to compute its affinity to that receptor to modulate its property. And this is the space where machine learning has made tremendous impact, especially because the one thing which we need for the process to work is the 3-dimensional structure of your target of interest. And over the past 2 years, Google, for example, came out with this technique called AlphaFold, which has completely revolutionized this field, because now you can generate the 3-dimensional structure of any target of interest through just its primary sequence. And AlphaFold team basically did a 3-dimensional structural elucidation for almost the entire proteome and released this to the public, which was a great service to the community. And the second big advantage has been these commercial libraries called Enamine from Ukraine. What they do is they provide us with a catalog of molecules that we can screen against any target of interest. And we give these companies a list of molecules which we think can bind to our target of interest, and they make and ship it back to us, which we can test. However, the chemical space is huge. And in the past 4 years, you can see that in 2019, to screen a 1 billion molecule used to cost us, in terms of dollar amount on an Amazon Web Service, around $12,000, extrapolated to 21, where we could screen 10 billion molecules for $120K, to 100 billion, where it becomes too expensive to do these computational calculations brute force. So what our group, as well as other groups, have tried to do is, can we use machine learning to speed up this process? And the technique that we are using is called active learning. So what actually happens in active learning? So what you do is you dock a random set of molecules into your receptor, say like a million molecule, which is computationally tenable, and then you build a machine learning model which understands the pattern with which these molecules are interacting with your receptor. And using that model, you basically pre-filter your original database to a small data sets that you can actually dock into your receptor. And doing this process, you increase the speed of this entire calculation 1,000-folds. And as such, now you can do ultra-large virtual screens. For a robust, it's possible even for a robust or for a modest compute resource that we have at Broad. And as a case study, I would like to show an example of an ATPase that we initially did not have initial 3-dimensional structure for it. So we could not have done any kind of hit finding for this particular receptor, but AlphaFold allowed us to basically generate a 3-dimensional structure of this receptor. And then we screened a billion molecule against this receptor, accelerated by active learning, to come up with a list of, say, 1,000 molecules, which we essentially, so here we are screening these large library of molecules against this receptor. So we come up with a small hit list, which we then provide to Enamine to purchase, and they were able to generate 80% of that, give it back to us. And we tested them biochemically, and we got some decent hits, like molecules which were 2.5 micromolar, 5 micromolar, and so on. These are not really drug-like molecules, because they need to still be optimized. But these were good starting points for this program, because we didn't have any success in any kind of HTS screen against this particular target. So that brings me to the next step in our process. let's say we have a hit. What do we do, for example, in the previous step? So we generally follow a DMTA cycle, which is design, make, test, and analyze cycle. So this is a circular process where we take our hit, make some modification to it with the help of a medicinal chemist, try to test it in our cellular assays and see whether there's an improvement in that process or not. And once, if we see an improvement, we update our model and we make further modifications. And molecular design essentially is a multi-parameter optimization. So we are trying to optimize for a molecule's biological activity, but we are trying to make sure that the molecules are non-toxic and more drug-like. So the two processes that we are using, first is the physics-based calculation on the right, which basically allows us to understand how a drug molecule is interacting with the protein. If you're making small modifications to it, how can we increase its target affinity? And on the right, we're trying to also optimize for a molecule to look more drug-like. So here we are using machine learning to try to predict molecular property. So I'll give a big shout out to Therapeutic Data Commons. It's a consortium coming out of MIT where they are trying to consolidate all kinds of ADME data for small molecules. So you can predict, using these data sets, you can predict molecular solubility, permeability, and its toxicity. So what we do is we take these models and we use a reinforcement learning loop. And it's an interesting machine learning kind of framework where there are two models which are fighting against each other. The first model creates molecules. The second model tries to rank those molecules, saying, oh, this is not a drug-like. So we penalize this molecule. So next time, do not create this molecule. So this constant fight between these two models result in this multiple parameter optimization, which is shown here. So as we are making modification on our molecule, our MPO score is essentially increasing. So in the end, what we end up getting is we are able to optimize for molecular affinity to a target, improve its solubility, permeability, while reducing its toxicity. And during this cycle, the molecule improves in these aspects, and we get more drug-like molecule. So this would be the end of mostly like lead optimization stage. And in the last part of my talk, I'll talk about where we can use computer-aided drug design or computer simulation to understand cancer resistance mutation. So for example, and I would like to give an example of Gleevec for CML, and this was a wonder cancer drug for the CML, and it was basically attacking the ABL kinase. However, a few years down the line, we started to get resistance mutation against this target. So what one of our collaborators did was he did a bunch of experiments on different mutants of this target and with imatinib bound to it or Gleevec bound to it, and tried to compute on the x-axis or get on your x-axis the binding affinity of the drug, and on the y-axis is how long a drug resides in the protein. And he was able to find out a particular mutation, N368S. And interestingly, this mutation essentially had the same target affinity to ABL kinase, but the half-life of the drug reduced by, this is a log basically reduced by almost like order of magnitude, or 3 orders of magnitude. So the molecule could bind tightly to the same target, but a mutant target, but was kicked out very quickly. So the question which we posed was that N368 mutation, which is far away from the binding site shown on the right, how was it affecting the drug residence into the receptor? And that's where these computer simulations are very useful, because this is more like a basic science discovery, because it's allowing you to make hypothesis and test hypothesis about what exactly is happening. So here we are using computer simulations to look at the dynamics, because proteins or receptors are not really rigid. They are molecules. They are living stuff. So they're actually moving in space. So we are trying to see how these mutations are affecting its dynamics. And we are using metadynamics. It's an enhanced sampling simulation to improve or enhance the dynamics of these proteins. But the problem is it creates such vast amount of data that humanly it's impossible to understand what exactly is happening in these simulations. So machine learning here is used to interpret the data which is coming out of these computer simulations. So what we ended up doing is first of all, on your right-hand side, we were able to replicate the experimental observed numbers, so that gave us confidence that we are in the right space. Our computation were agreeing with the residence time that we got from experiments. But more importantly, we were able to also come up with a hypothesis of why a mutation is reducing drugs' residence time. Why is the drug not that effective with this particular mutation? And what we found out was there were two pathways for the drug to be released in Abel kinase, wild type versus N368S mutation. And wild type, in case of the wild type, if in this animation showing, you can see that the drug is actually sticking more to the target and is coming out from the back pocket, which is slow. But what actually this N368S mutation did was it made parts of the protein very flexible, and that allowed the drug molecule to get out from the front end of the protein, which was faster. And using that, we were able to explain why N368S mutation was resulting in a lower efficacy for the drug by decreasing its residence time. And based on that, we were also able to use a mutual information algorithm to come up with whether there are other mutations which can do the same process. So long story short, these kind of simulation actually also helps you to understand the mechanisms of resistance mutations, which are happening at points on the protein or receptor far away from where the drug resides. So finally, I would like to thank my organization, Center for Development of Therapeutics. So we have a group. So I'm part of the molecular design group, but we have a very collaborative environment where we have structural biologists, computational chemists, as well as a bunch of biophysicists working together in the early stage drug discovery, and also my collaborator, Dr. Tiwari from University of Maryland, and my collaborator and mentor, Dr. Singaroy from University of Arizona. So thank you for listening to me, and love to answer your questions.
Thank you, Dr. Shekhar, for that great talk, as well as Dr. Chapiro and Goeppert for theirs. While we're waiting for folks to come to the audience for questions, I'll kind of get things started. Given that we often will need next-generation sequencing, which will come from a biopsy, aren't all roads going to really lead to pathology? And in a biopsy. I ask this question because I'm thinking about the evolution that we're seeing within hepatocellular carcinoma, where we've relied upon a LIRADS diagnosis for so long, and we're recognizing that a lot of those, even meeting that criteria, turn out to be something else, often intrahepatic cholangio, which remains a very challenging thing to diagnose in many centers for multiple reasons. So I'm trying to think of a way in which AI radiology would lead to a path where there would not be a biopsy involved.
Well, we are having very similar conversations on all HCC conferences where for the longest of time we've been using LIRADS 5 and not biopsying a lot of the tissue. And that was possible, and that was explained for the sake of non-invasiveness, and obviously cirrhotic patients tend to bleed, and then transplant risks and seeding, et cetera, et cetera. And now we're looking back at 20 years of wasted opportunities of not having tissue and not having the ability to go back and bioprofile a lot of the tissue that we need to develop new drugs and to understand the outcomes and long-term outcomes of patients. I think that radiology can provide very valuable holistic characterizations of the tumors, when biopsy can't, because you always take just a small little piece in light of tumor heterogeneity. But I do think that there is no way to avoid tissue sampling. And from my perspective, I just don't see how that's going to be a responsible strategy, on molecular targeted therapies of the future. I mean, I'm not sure if you agree.
Yeah, I totally agree. And I think this story with the HCC, I mean, this is, as most of you know, this came from the Jordi Bruix and Barcelona trial. These were certainly expert radiologists in the HCC field, but it reminds us again that guidelines have to be for the world, not for one center. If you do a guideline for a world and you prove it for you, then it will, at the end, maybe not come out ideally for all of us. Yeah, because the radiologists are not the same everywhere. Pathologists are not the same everywhere. You have to do guidelines. If the guidelines are for the world, then you have to do it for the world. Yeah, and this is the first thing. And the second thing is, as you mentioned correctly, at the end, you mostly you need some, or you have questions later on that you initially didn't have, and you think, oh, it would be great if we would have this tissue, yeah, and it's quite easy to perform a biopsy on the liver. So I think we should go for this. Yeah.
Microphone 1.
Thanks. My question is for Dr. Shekhar. Thank you for your great talk. I'm trying to understand the scope of the targets that you can model. And so a lot of the targets are in membranes or they're in phase separations. And I was wondering your ability to model those more complex interactions.
Hello?
It's on.
Yeah, so are you talking about membrane proteins, like the proteins which are embedded or targets which are embedded in a lipid bilayer?
Yes, and also the proteins which have the unstructured domains that form these condensates. Yeah.
Right, so for unstructured domains, it's become slightly difficult because we are working with a three-dimensional structure of the protein. So these intrinsically disordered proteins becomes a target for, becomes a difficult target. But that's where physics-based methods can be more useful. The second is you're talking about, say, for example, proteins which are embedded in, as like, for example, membrane transporters or ion channels. Yes, those we can use these methods for those as well. That shouldn't be a problem.
Right, thank you.
Excellent. It looks like we have someone in a green jacket, importantly, microphone number 4, sure. So you get priority over Dr. Valle for sure.
Hi, I'm really passionate about getting diagnoses early and in communities. The people at the large centers, you know, there's multiple radiologists. You guys can sit in the dark room and talk about it and probably come up with the right diagnosis most of the time. I'm wondering how long it'll take to get to Petoskey, Michigan, to the small hospital I work at out in north of Detroit. How long does it take to get to those places so that, and I'm also interested, how long before you're able to like read CTs where the internist didn't order contrast, you know? Are we gonna be able to do that? Because I think the pathologist, I can't see your name, so please forgive me, but you know, you pointed out for the experts, AI was equal, but it's for the non-experts that I think this is maybe gonna save our lives.
Maybe I can take this one with the caveat that I'm an interventional radiologist and never sit in a dark room. But other than that, yeah, so I think we, at this point, have very powerful clinically deployed software that supports us already clinically. For example, as I was sitting here, my phone rang with a message from software called AI Doc that informs me that in one of our hospital emergency room scanners, there is a patient with a pulmonary embolism. And I know this message before the diagnostic radiologist even has the scan in his line, all right? So that is a common pathology that is relatively easily deployed. That system is available now in many practices. Whether or not we're going to be able to do something like that for liver cancer or cholangiocarcinoma specifically depends on how well we get organized in terms of our data. It's technically doable, but we need to make the right effort to make it possible. In terms of your second question, for example, teaching trainees or improving image quality, that is being currently logistically deployed by Siemens, Philips, GE. We're applying these things. We're improving image quality, reducing dose. This is technology that is not futuristic. It's there and it's being applied. We're using it on a daily basis. So I'm optimistic. The only concern that I have for this particular pathology of cholangiocarcinoma is the paucity of data and the sort of right now global lack of organization. At the end of the day, it's a rare disease, and it makes it really hard to tackle, right? So that's my
As a
comment.
follow-up and an intersection to that question, put towards Dr. Goeppert as well, is one thing that we often see is patients have a biopsy taken of their liver, and it's sent someplace because of hospital agreements that may not have the capability to actually make a diagnosis. They exhaust part of the sample, and then when the patient, then we get that sample back because insurance isn't going to pay for another biopsy, now it's this cycle that we get into, and I think it's a big part of the delay. So is there an opportunity for AI radiology to then help inform where the pathology specimen goes?
Yeah, so so one of the drawbacks in the United States as opposed to some countries in Europe is that we don't have a centralized EMR system, but Epic is probably the dominant system, is working overtime right now to exactly make sure to develop these workflows where a sample goes to the right person with the right specialty, with the right emphasis, and that for example at Yale we build on subspecialization. We have liver pathologists. That's not something that you have everywhere in every institution, but we have dedicated liver cancer pathologists that read all such studies, trying to reduce the variability of the reads and making sure that the entire group of patients in Connecticut is given the best care. So I think it's where it's going. In cholangio, the paucity of data will make it the lower priority target for these big companies like Epic. But we're developing at least the infrastructure right now on all frequent pathologies to afford us exactly that, right?
Thank you. Juan, on question one.
Thank you. In clinical research, of course, we try and standardize as much as possible the data field so that we've got what we think is a good readout once we get the results. So my question is, in terms of pathology or imaging, how much does the process have to be protocolized to have standard imaging? In other words, can you work with dirty data, scans that are not quite per protocol, thick size scans, maybe immunohistochemistry that's not got quite the right staining? How much correction can be made for things that are not standardized?
Yeah, for the pathology part, I can answer H&E is a quite standardized stain, so this is not too hard to accomplish. And the immunohistochemistry part, at the moment, I'm not aware of studies that using immunohistochemistry. They start all with morphology, and that's not, from my perspective, not a bad thing to do, because you start with the morphology, and then as I could show to you, hopefully, a lot of immunohistochemistry is becoming useless. And this is a good point, because you can use the tissue for other things then. Yeah.
Excellent. I think we have Dr. Javli at microphone number 2, I believe.
Yeah, thank you, Skye. First of all, this was such a wonderful session, so congratulations to all. My regret is this is one of the last sessions, so most people have left. But, in terms of practical questions for cholangio, one issue we face is the accurate diagnosis of perihilar cholangio, large duct cholangiocarcinoma. Even the diagnosis of intrahepatic cholangiocarcinoma. The average time from symptom to treatment, according to the foundation, is close to, I think, 8 or 9 months. So I think the kind of wonderful tools that you described, using radiology, using pathology, in order to make the diagnosis in a more succinct and earlier way, would be a direct line from what you have done in your lab with what we can apply in the clinic.
I mean, I'll second that. I mean, I think perihilar is where we have so much work to do in diagnosis, care of patients. I mean, as we've talked about, it represents the lowest cohort within our large trials, but the overwhelmingly largest proportion of patients that have a biliary tract cancer. So a lot of challenges. Dr. Chapiro?
Yeah, so I actually, 2 days ago, I did a case exactly like that, a new patient with central mass needing an internal, external biliary diversion, and then I also did biopsy that mass internally and was able to hopefully diagnose it today. I just received a message that we were. But these are very hard to diagnose, and I think before we apply these super aspirational technologies to a very small cohort, I think what we need to do is we need to educate people who actually diagnose these cancers. And my personal, what bothers me is that we have a group of 19 interventional radiologists, right? And this patient came to me by pure coincidence. And I know that, for example, in at least like 15 out of the 19 colleagues in my group would not have known or had interest or been educated enough in topics of liver cancer to actually do it exactly the same way. And then the patient would have dwelled another month, et cetera. So I think that in order to tackle these situations, we really need to educate people on cholangiocarcinoma. And I think that's a much bigger issue. And I don't think that AI will resolve that. I think we need to invest a lot more in education.
Microphone number 4.
Yes, thank you. Just a wonderful, wonderful conversation, and it's so exciting. I guess I had one quick question. All of you up here and your institutions are driving the science and the medicine and the solutions in terms of being able how to use AI and machine learning to take it to the next level. I guess my only question is how important is Google and NVIDIA and Microsoft and your kind of institutions, joint ventures with them, taking it to the next level? There was a comment about the technology is here. So is the ball now all in your court, or how are you trying to direct Google and NVIDIA and Microsoft to improve the AI to help the entire situation?
Dr. Shekhar?
So, yeah, I can use this. So, for example, Google and NVIDIA, like I can give an example for Google. So, DeepMind, their engine was able to get the AlphaFold model done, and that enabled a lot of virtual screens to be done by us. So that was a big impact which was made by Google. NVIDIA, on the other hand, is providing compute resources in terms of getting the latest GPUs available either through purchase or getting through through a compute grant. So yes, we make, we can run our models on their resources, but as such, NVIDIA doesn't build its own, from what I understand, at least for the drug discovery, I may be wrong, but I don't think they're doing anything like model building as such. So we use their hardware, as I would say, to do the calculations.
I would add that all these companies are not really interested in a disease like cholangiocarcinoma, which is to my frustration. When I approach them for HCC, which, as you know, is much more frequent, they're not interested in this. They're interested in lung cancer, in breast cancer, and in really highly data-rich and prevalent environments. They want to have hundreds of thousands of patient data points. When we approach them with HCC, they're like, well, this is like maybe like what, like the 8th most common cancer? Let's just tackle number 1 first. So, that's how they think. They think about the low-hanging fruits first, data plurality before, a tight core. But they have an important function. I mean, they design and build pipelines that we can then use and reproduce, and that has a downstream effect on us. But, when we approach them with interest, they're like, well, you have like 1,000 patients. What am I going to do with this? This is not valuable to them, unfortunately.
We'll take our last question on one, and I think for this, we really need to deploy some epidemiologists to tell them about the wave of primary liver cancer that's coming. So, MAFLD is, it's here.
I agree.
So, question 1.
You just went a long way toward answering what I wanted to ask. Let me put it this way. We have available very inexpensive neural network machines that can be fairly straightforwardly programmed to analyze images and pick out features that meet some criterion. And even if the cheap ones aren't up to the task, there are online or could be set up online systems to do that kind of thing. In terms of access by remote facilities that aren't equipped to do the ideal radiology interpretation. Would it be practical to have specimens prepared at local hospital sites and images acquired? Are they capable of getting good enough images that you could simply send them over the internet to a big machine that could do the analysis and send back the results?
Absolutely. I mean, the penetration of really good hardware and good imaging being acquired in the periphery practice groups is not bad. I mean, you know, I wouldn't say that we have a lack of data or that everything that isn't acquired in an academic center is poor data. I think it's just the centralization that we're missing. And if we, for example, were to develop a portal centrally where one can submit for, you know, for centralized storage, I think people would do it.
So that could be a task for foundations that are concerned about rare diseases, for example.
Absolutely.
Yeah.
Yeah.
Well, thank you very much for everyone for staying. This was a fantastic session. Thank you.
Okay, a few closing words before we lose the last of the diehards here. So it's been a very exciting 3 days. You know, if I were to choose the words, it would be I was enthralled, exhausted at times, but leave excited and energized after hearing all the work everyone is doing, all the collaborations that have occurred at the meeting. And it has been very, very critical for the patients, their families, friends, and caregivers to have been here with us as part of this journey. We heard a lot of inspiring stories today from patients and caregivers. I saw James Hastings is still there, and he reminded us of skanxiety. I hadn't heard that term in a while. Jules Wish, Honey Hirame Smiler, who all spoke with courage and shared their stories, and we're very appreciative of that. The poster session was a great success. We even had an update on the TOPAZ study for three-year data. Usually this would be presented at our more legacy meetings, but it's clear this meeting is becoming the place to present and discuss research relevant to cholangiocarcinoma. So word of appreciation to AstraZeneca and their leadership for making this meeting the place they wanted to put that data out. We heard a lot about different difficult topics, and that's why the sessions all had these kinds of themes: global challenges and global solutions, where we had speakers from 6 countries coming from thousands of miles away to talk about the barriers they face, but also the innovative solutions that they've come up with. And hopefully as the years go by, we will see less and less barriers and more solutions. Rational combinations of therapies. We know one drug at a time is probably not going to do the trick, so that group of speakers did a tremendous job in tackling this very difficult challenge. This morning, it was an act of God probably to get regulators from all these agencies and countries to come and be in one place with academic investigators, probably much harder than arranging a G20 or G7 summit. And lastly, AI and machine learning. Usually, you know, people disappear for the last session, but I think the ones who left were the ones who truly missed out. That was an outstanding group of speakers with application of, you know, what are technologies that will likely change the world. So we had a lot of hugs, a lot of hope, but most importantly, a lot of action and collaboration while this meeting ends today. We know a lot of work will happen between now and the next meeting, so look forward to seeing you then, and I'll hand it over to Stacie.
I want to extend, let's see. I'll just move on. We'll come back. Oh, there we go. Okay. I want to just extend heartfelt thanks to all of our chairs, especially to Mitesh. Mitesh has been a rock for this foundation. He was here at the very first meeting. He was the one that conceived of the ICRN, that we needed to do a research network. He has been, we text a lot on the weekends. Every time he sees something exciting, I get a text, emailing. These investigators have spent thousands and thousands and thousands of their hours helping us put this organization together. So really, so truly grateful. And they did an amazing job with this conference. So just so much thanks for them. Also, so grateful for all the speakers, the poster presenters, and all of you attendees. I want to also give an enormous thanks to our sponsors. Their support makes this event possible. I especially want to thank AstraZeneca and Inside, our Redwood level sponsors. We're just thrilled to have so many companies interested in this space, interested in developing drugs for the patients. I want to also just remind you that next year, April 9th through the 12th, the conference will be here in Salt Lake City again. The chairs will be Dr. Nilo Azad, Dr. Ben Stanger, Dr. Chiara Bocconi, and Dr. Rocio Rodriguez Macias. Remember that this is the Research Priorities Consensus Conference. It will be formatted the same, but the presentations will be a little different. I think it's going to be very exciting to be a participant in this. So please put it on your calendar and please come. We will need you here. This is going to be an opportunity for us to identify the research priorities across the globe, across the multidisciplinary spectrum, and get aligned on the available research funding and leverage additional funding from diverse sources to accelerate breakthroughs in this research. So I wanted to just end with our refreshed logo and our new tagline, Strengthen Community, Hope, and Research. I hope that's what you felt here today. I want to let all of the attendees know that you will be receiving a survey by email after the conference. We'd be so grateful if you would fill that out. Your feedback helps us with the next conference. I also want to let you know that our media partner, Amplity, is hosting a networking event from 5 to 7 p.m. here at the Salt Palace. They'll have kind of just light refreshments. It's in 250A, and Dr. V. Sahai, and Kelly Butler, who's a research advocate for CCF, will be sharing just an overview, kind of the hot topics that we saw. It's kind of fun. It's live broadcast. It's fun to be in that room and watch them do this. So you are welcome to do that. On a personal note, I just wanted to say thank you. It's just a pleasure to meet so many of you and to be all together in this room once a year. This year, a great movie came out called The Boys in the Boat. And if you didn't read the book, you really should go back and read the book. I read the book when it first came out years ago, and I was so inspired. I was so inspired by the resiliency of the human spirit, and by what a group of passionate people who have a singular goal can do together. So I just wanted to say thanks for being on this boat. We'll see you next year.